I'm Kourous Rezaei, Chief Medical Officer of Ophthotech. Ophthotech has gone through a transition to be a gene therapy company in the past 18 months. And we are focusing mainly on retinal diseases. And you may ask, why doing gene therapy in the eye? The eye has the advantage of allowing you to do local delivery to the tissue of interest without bypassing any systemic side effect that usually you conquer with systemic treatments. The eye is also relatively immune privileged, so you may have a limited amount of inflammation after gene therapy. And it allows you, by manipulating the vector or the promoter to be tissue specific, to limit off-target implications of gene therapy. Can I have the next slide, please? You have the. These are my forward-looking statements. And this is a brief overview of our pipeline. Our lead product is rhodopsin-mediated autosomal dominant retinitis pigmentosa. This compound candidate will go into trial next year in 2020. Our next gene therapy candidate is for Best disease, which will go into patients in 2021. We're also working on mini gene therapy for LCA10 due to CEP290 mutation, and also for Stargardt disease due to ABCA4 mutation. We also have a therapeutic pipeline for Zimura, which is a complement C5 inhibitor. We are reporting phase 2b trial results later this year. And we're also assessing Zimura in autosomal recessive Stargardt disease, which we will be reporting next year. The enrollment for both these trials are completed. We're also working on an extra 1 inhibitor, which we will file IND in late 2020. Thank you.
Thank you very much for that. And I guess you sort of answered the question at the beginning, but maybe you could go into a little more detail in terms of the eye as a focus area, and sort of how you might expand the pipeline into potentially other tissues and going forward.
Sure. So again, in the eye was one of the organs that you can actually see the tissue directly. Surgically, you have access to get, as I said, we are working on retinal diseases, so you can actually gain access to the subretinal space surgically to deliver the vector to the area of interest without exposing the entire eye or the entire body for that effect. And going back on the foundation of our pipeline was based on strong, compelling science, significant unmet medical need, and financial viability to create value. And any of those indications that I pointed out to you, currently there are no treatments available for patients. Regarding our lead product, for rhodopsin-mediated autosomal dominant retinitis pigmentosa, it's a very elegant compound candidate because in these patients, you have a mutation in rhodopsin gene that the patient produces toxic rhodopsin. So what you need to do is you need to knock down that toxic rhodopsin and replace it with a healthy protein. This compound candidate does both. It shuts it down with a short hairpin RNA, replaces it with a transgene that produces the healthy protein, both in a single vector, and it is mutation independent. Autosomal dominant RP has over 150 mutations in around 10,000 patients. This product candidate is mutation independent, and will be going into patients in 2020. Regarding the autosomal dominant BEST, again, the patient population is somewhere around 10,000 patients. And in the autosomal dominant BEST, you have a haploinsufficiency of that not enough protein being produced. And beauty of science on both autosomal dominant RP and the Best disease that I just talked about is that the proof of concept was done in naturally occurring dog models. So the autosomal dominant RP, they looked at the mouse model, which is usually a knockout, but the natural occurring dog model is actually a dog that gets the retinitis pigmentosa or a dog that gets the Best disease. And they were able to show long-term we were able to show long-term functional and anatomic rescue in both these indications.
The mini gene program is interesting. Can you talk a little bit about that and how that's developing?
Sure. So obviously, AAV, or adeno-associated virus, is the ideal vector for the delivery of the transgene because already, for example, Luxturna is already FDA approved. We have safety information about the virus. However, the limitation of AAV vector is the size of the transgene, how much cargo you can put in. And for example, LCA10 is the most common LCA due to mutation of CEP290. However, the size of the transgene is just too large to be able to be put in or fit inside an AAV vector. And what the scientists have done in our group together, we're collaborating this project with UMass and Horace Gene Therapy Center. And what we've shown is that in the CEP290 knockout mouse, which is a very aggressive knockout, you put a fragment of that gene in the vector and transfect these mice at P0, and you're able to have a rescue at 3 weeks. Basically, by 3 weeks, all the retina is almost completely degenerated, other than the area that was transfected. And this is, again, a mutation-independent concept, but the idea is that you use a fragment of the gene that is still able to produce functional protein. And we're deepening our relationship with scientists at UMass. We started with LCA10, and we're going to update the public later this year on how we're going to move forward. But we're also expanding that also in autosomal recessive Stargardt due to ABCA4 mutations.
Yeah, so obviously the eye is a competitive focus area for many companies. And I think the mutation independence component is a differentiator. Maybe you can just talk a little bit about the sort of competitive landscape and in terms of the 3 programs you just discussed, what's different?
Absolutely. As you know, in orphan, you need to generally be first. So for autosomal dominant RP, we are not aware of anybody currently being in patients doing the gene therapy for either of them. Regarding CEP290, again, being a one-time delivery and being mutation independent covers the entire spectrum versus, many of, especially CEP290, if you do it in the children or a younger age, it's not going to be that easy to do frequent injections. So we believe that it's in a one-time cure may be a better answer. And again, there are other technologies out there, but we believe, to my knowledge, this is the only one that a rescue has been shown in a mouse model. And just a little fine point about the mouse model, when you inject a mouse, to my understanding, at P0, you basically have only cones, and cones are only 5% of the photoreceptors. And if you can show rescue at that young age, that's a pretty strong scientific evidence.
So could you just, you mentioned some updates you're going to give to investors. Maybe it would be helpful just to recap from a pipeline perspective what we should be looking for this year or next year in terms of data and updates.
Sure. Regarding our gene therapy pipeline, we're going to update you later this year regarding how we will be proceeding with our LCA10 due to CEP290 mutation. Next year, we're going to be going into patients for rhodopsin-mediated autosomal dominant RP. We have a clear path to IND for that indication. Currently, we are doing IND-enabling studies and natural history studies. In 2021, we will be going into Best disease, again, doing IND-enabling studies and natural history study disease. And then regarding our therapeutic portfolio, we have a complement C5 inhibitor named Zimura. We are doing a phase 2b clinical trial for geographic atrophy due to dry macular degeneration. Results of that trial will become available later this year. We're also doing, again, a trial in Stargardt disease with the complement C5 inhibitor Zimura, where the results will become available next year. Both trials obviously are fully enrolled.
So one of the themes that we've heard today quite a lot is just manufacturing and how you're thinking about, obviously the gene therapy programs are sort of earlier stage, but how are you thinking about building out scalable manufacturing, and are you thinking about working with partners or doing that in-house?
That's a great question. Manufacturing is a key component for gene therapy, also from the financial perspective. We have not publicly announced at this point how we're going to move forward. Obviously, we have planned it since we're going to be in patients next year. But in ophthalmology, again, one of the advantages is that you need a very small, the amount of gene vectors you need is limited when you compare it to, for example, if you're treating systemically. And when you're getting started, again, you always look at whether you want to contract it outside or having it in-house. But again, based on what I said, we have not publicly announced what we will be doing, but we have already have plans moving forward. But it's a great question because CMC is a key component.
And then what about moving forward in terms of, obviously you've got a lot going on already in the AI program. Are you thinking about moving into other areas that are also immune privileged, for example? Or how are you thinking about long-term research?
It's very interesting because if you look at, I just came across this graph, there are over 250 retinal diseases with monogenic mutation. So far, we have only one treatment FDA approved. So obviously the field is wide open. And we still, I think we are scratching just the surface. And our BD team is still actively looking based on the three foundation factors that I just mentioned: compelling science, significant unmet medical need, and commercial viability. And if we come across any of an asset that has those characteristics, we would be interested.
And just in the last few minutes here, in terms of the actual AAV vector, are you working on anything there internally in terms of engineering, or is that, I don't know what you've disclosed on that front?
Yeah, we have a collaboration with UMass and Horae Gene Therapy Center where we work with mini gene therapy. At the same time, one of our collaborators is Dr. Gaoping Gao, who is a pioneer in AAV vector technology. He was actually, I think, the first author in the filing of the IP from UPenn. And we are looking at we are doing a comprehensive study at different AAV vectors, both intravitreal and subretinal application in the eye. And based on that information, we can see which route is the best route for any future disease. For example, we're looking at Stargardt. We want to do it subretinal, intravitreal, and which vector we want to use that gives us the best expression. And that study is currently ongoing.
So you sort of have access to doctors. He has quite a few patents, I believe.
Absolutely. You know, we at Ophthotech do not have laboratory. Our scientific horsepower and talent comes with our collaborations with universities. We collaborate with UPenn, UFlorida for ADRP and Best disease. One of the things we do strategically is not only to acquire the asset, but also build a deep relationship with the scientists. And that's why we started with ADRP, then we moved to Best. We started with LCA10 at UMass, and now we are moving to Stargardt disease and also doing the vector comparison between different AAV vectors.
Great. I think we're out of time. Thank you very much.
Thank you for your time.